the first thing we need to do is called the polymerase chain reaction. The polymerase chain reaction's whole purpose is to amplify DNA, make more DNA, make lots of copies. Okay, and so PCR is essentially needs a DNA template, needs primers, needs DNTPs, some salts and other things, and a DNA polymerase. What does this sound, what physiological process does this sound just like? Now, if we think about molecular techniques, they're all based on some physiologic process. The one that PCR is based on is, yes, wait for it, DNA replication, right? DNA replication requires a DNA template. It requires primers. In the case of replication, they're RNA primers put on by primase. In the case of PCR, we use DNA primers that we manufacture or we make or order from a company. Okay, and then DNTPs, we know those are just deoxyribonucleotides. The reaction buffer are salts and other calcium salts, magnesium salts, other things that help the enzymes work, and DNA polymerase, just like DNA replication. So again, it's a DNA polymerase, which means it has to obey the rules. What are the rules of DNA polymerase? Yes, juicy 3' OHs can only add there. Everything has to be anti-parallel and complementary base pairing. Yes, my favorite things ever. All right, so primers are required to run PCR, to do PCR, to make polymerase chain reaction happen. Of course, this figure from the textbook, from our 107 textbook, is wrong, and I think this is an old version, so maybe the current version is better, but whatever. The region of DNA to be amplified by PCR or to be copied is actually from the end, the 5' end of one primer to the 5' end of the other primer. This would be the entire region that gets amplified. And so one of the primers needs to be complementary to one of the strands, right? And the other primer needs to be complementary to the other strand. So the primer sequence that they're showing here, up on top, that would be the 5' end of the primer, the 3' end of the primer, and it sits on this strand. This primer down here, this is the 5' end of the primer, this is the 3' end of the primer, and it sits or is complementary to that strand. Okay? And so it's complementary to that strand, and this picture's a little bit difficult to understand, but that's what the primers are like, which means if it's complementary, what does the base that goes with G have to be? C, T, A, C, 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 A, T, etc., etc., etc. Complementary base pairing, right? Polymerase will grab onto the 3' end and start complementary base pairing to this template strand going that way and to this template strand going that way. Okay, so let's talk about the process. This is just a picture of the primers. So these figures are a little bit better. So first we have to start with our template DNA, right? So whatever we isolated, we pulled a piece of hair out of somebody's brush and it has that little skin tag, and we isolated DNA from that. We've synthesized our primers or ordered the ones. We have to know the sequence of the target in order to use PCR, right? The template DNA sequence, in order to make primers that are complementary to it. And then we need a whole bunch of nucleotides, deoxyribonucleotides. We first have to heat up the double-stranded DNA to separate it, right? Breaking hydrogen bonds, just like in replication, but in DNA replication, what does it? Helicase. In the case of PCR, we just use heat, so we don't need to use another enzyme, right? It's much simpler that way. So we have the single-stranded DNA, we have our primers in there, right? The next step would be primer annealing, and that just means complementary base pairing to the template. Complement to the template. And so when we go from really hot up here, and then we cool it down a little bit here, that allows these little primers to sit or to a complementary base pair to their target sequence. Okay, and so if this strand here is 3' to 5', then this guy has to be 5' to 3'. We'd add to this end, and it would fill in that way. 5' to 3'. When polymerase grabs on, it'll fill that way, adding to 3' ends, right? Always in the 3' direction. Okay, and so that's what they're showing here with these dark colored guys, right? That's extension. That's when DNA polymerase is active. Whatever temperature your DNA polymerase is most active at, that's where you'd set that temperature. And we are just doing complementary base pairing. So it's just like replication, right? Has to start with a primer, needs that juicy 3' OH, and then can extend. So here we go. These guys have extended. And then we just do this cycle over and over and over, which means we'd have to heat them. These two things would separate, right? And then the primer could sit back down on this template or sit on that guy. These two would separate. The primer could sit back on this guy or sit down over here on that guy, right? And we do this over and over and over and over. And if we repeat this 20 to 30 times, we can produce millions of copies of the DNA between, right, primer to primer. Okay, so all the way from that and all the way from this, and then they'll reanneal coming back together as well. And that, my friends, is PCR. What they do in order to allow for this heating, right, when we did the beginning, when we heat to denature, to break the bonds, break hydrogen bonds, well, you can't, that's boiling, essentially, or almost, right? About 95 degrees Celsius, 100 degrees is boiling. So that breaks all the hydrogen bonds, but it also kills all enzymes, right? But Taq DNA polymerase is from a bacteria that's a thermophile, which means it's one of those bacteria that lives in the ocean floor hot sea vent where it's boiling water down there. So this enzyme does work at very high temperatures. So that way we can go through this cycle of heat, cool, extend, heat, cool, extend 20 to 30 times, and it does not kill our enzyme. So we don't have to add enzyme at every cycle like they used to have to. Woohoo, graduate students. And then here's just an overall better picture of the polymerase chain reaction. And if you only have a teeny tiny amount of DNA, you really only need a couple molecules in order for PCR to work. That's the power of PCR. So you can have a teeny tiny amount of the target, your primers, right? Add to 3' end, add to 3' end, denature, reanneal, extend here, right? Denature again, reanneal, extend, and so on and so on and so on, until you get millions of copies of the target sequence that occurs between, right, the two primers. That sequence right there. Okay, so here's another picture. Like all polymerases, just to remind you of the rules, Taq polymerase can only add to 3' end, 3' OH of an existing nucleotide, right? So if this is our template and this is our primer, there's our template. Our primer has to be 5' to 3', right? Has to be anti-parallel, and we're going to add to 3' ends. That means this guy has to be 5' to 3'. This guy has to be 3' to 5', add to 3' ends. We'll get two identical DNA molecules formed, right? And we need to have that primer, just like in DNA replication. So be prepared for questions comparing PCR to replication. One of the main differences, well, PCR is done in a test tube, and DNA replication is in a cell. DNA replication does all the DNA. The polymerase chain reaction only amplifies what's between two primers. The primers for DNA replication are RNA primers. The primers for polymerase chain reaction are DNA primers. We could make RNA primers, but we don't because it's more expensive and they're not very stable. So we just make DNA primers and it works just as well. All we need is that juicy 3' OH to make this happen. All right, now we're going to do some practice. So I'm going to walk you through how to design primers for PCR. And then in the next slide, you're going to do that for me and do a screenshot and an upload. So let's say we want to amplify the entire region shown. Where do we make our primers? That's question number one. And then what is their sequence? Again, just like we did for DNA replication when we wanted to sit primers down at the origin, it was a little bit of trial and error. You can kind of do it in your head because you know we have to add to the 3' end. Right? Our primers need to go 5' to to 3', and then this is the direction that we need to amplify, right? So this little primer sequence can't be at the end and this falling off. We want, right, we want to get all of this sequence between here and here, which means we'll want to sit a primer over on the left side, and we want to sit a primer over on the right side. So we can see that it needs to be, right, the rules anti-parallel. And so if we want it to go in this direction on the left-hand side versus the right-hand side, right, we want our primer to sit going this way, right? So the first thing we're going to do is we're going to go 5' to 3', right? And then we can add, right, then we know we'll add in this direction, okay, because we want to get this whole strand going this way. Doink, 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 right, over to there. And on this side, we know it has to be anti-parallel, and we have to add to 3' end, so it needs to be going in that way, so we can go this way, right, doink, 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 all the way over to the end. So what does that mean? What does this line mean? These are deoxyribonucleotides. They need to sit down. When we say sit down or anneal, what do we mean? We mean complementary base pair. So this little stupid line needs to be complementary to its template. Okay? So what's complementary to T? A, G, C, T, A, T, C. And then polymerase, this would be the primer that we've made, right? If that's how big we made our little primer, here's our juicy 3' end. Polymerase would come in and continue complementary base pairing, right? G, C, 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 G, 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 T, 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 blah, 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 and so on in that direction. Does that make sense? That means our other primer must be complementary to this strand as the template. Right? Just like in DNA replication, both strands serve as a template strand for PCR and for DNA replication. So this guy would have to be G, G, C, G, A, T, T, A, C, G. And his 3' end sticking off of that, that would be the sequence of our little primer. And then Taq DNA polymerase would grab onto that juicy 3' OH and go G, T, A, C, G, G, etc., etc., etc. So what are their sequences? If we were going to write them out, and we always write things out 5' to 3', the left primer would be 5', A, G, C, T, A, T, C. And you only need to make 8 or 9 base pairs. And on the right, again, we always list them 5' to 3'. So this one's going this way. So we'd have to go 5', G, G, right? Because we're reading backwards. C, G, A, T, T, A, C. I guess I still had a G in there. 3'. That's how we always, always, always write out our primers. We never write them out 3' to 5'. That's just not conventionally how it's done. So you will be asked, what is their sequence? You must write both of them 5' to 3'. The best thing to do is to do it exactly the way I showed you. Set it up on each of their templates. Write them out so that this one is 3' to 5' first. Then do the rewrite, I shouldn't use the word transcribe. Then rewrite this guy, right? This G is that G, G, C, G, A, yada, yada, yada. Is that clear? Great. Good job, people. Okay, now here's yours. This is the one that you're going to screenshot and upload to the PCR homework on Wednesday. We want to amplify the entire region shown. We want to add primers to the appropriate locations. Please label your polarities. Draw an arrow in the direction a DNA polymerase will extend. Okay? Add the primers to the appropriate locations. And when I mean that, not a line, not lines. Use DNTPs. Okay? Use complementary base pairing. Do it exactly like you're supposed to. You You can just write them next to the sequence. You don't have to rewrite them out 5' to 3' if you don't want to, but it would be really good practice if you did. Take a screenshot and upload.